Hello, hopefully this is working now, and welcome, and thank you for joining us today. My name is Dr. Brad Bowman. I'm the Chief Medical Officer at Healthgrades. I attended medical school at the University of Washington. I went on and trained and received board certification in internal medicine. I practiced internal medicine for 15 years, and in the 90s I started a company called WellMed. WellMed was one of the first online population health management companies. I later sold that to WebMD, where I then was the senior vice president for clinical innovation for over 10 years. For better or worse, I'm probably best known for creating the original WebMD symptom checker. So the format for today is I have a series of questions that I've been asked over the past several weeks. I chose these questions, one, because they're really great questions. And they're important about kind of aspects about how to managing COVID. I also chose them because many of them serve as kind of a jumping off point where we can get into more detail about COVID. So the first question I'd like to kind of go over today was one, I would like to know if this is really a physical virus that is attacking the world. Have you seen it under a microscope, in blood work, lab research, etc.? So short answer to the question is, yes, this is a real physical virus, and we actually know a lot about it. So just to begin with, when I first heard this question, I thought it was kind of an odd question, and then it kept coming up. I guess what I should have realized at the time is that, we don't talk about viruses a lot, or at least we don't talk about viruses in a lot of detail. And certainly COVID isn't your usual virus that we experience in sort of our day-to-day life. So, short answer, yes, this is a real physical virus. We know a lot about it. And to start off with kind of viruses, the first thing to know is viruses are really, really small. Viruses are, kind of, 1/100th the size of a bacteria. And bacteria are much, much smaller than regular, even sort of human cells. So when we talk about have we seen it, it's yes, we've seen it, but they're so small you can't see them with a regular microscope. You actually have to look at them with what's called an electron microscope to sort of see a virus. And then when we look at sort of viruses that way, if you were to kind of cut it in half and look at it in cross section, what you'll see with this COVID virus is there's these little structures, we'll talk about them more later, called spike proteins or flowers kind of on the surface of it. And when you look at it kind of in profile, those little spike proteins actually look like a crown, which is where the coronavirus or crown, corona for crown, actually got its name. So in terms of what we, talking about viruses, they're super simple structures. They really have only three main parts. So to begin with, think of a virus as like a tennis ball, like a really super small tennis ball. And on that tennis ball, there's these little flower spike proteins. They look kind of like bean sprouts, kind of like mung beans, kind of just sticking on the outside of the tennis ball. And then rattling around inside the tennis ball is some genetic material, either DNA or RNA. Now with coronavirus, it's RNA. So if you remember RNA, it's kind of related to sort of DNA, only it uses a uracil instead of a thymine to attach to adenosine. So thinking back to high school biology, that sort of stuff isn't important other than just know that there's RNA viruses and there's DNA viruses, and this is an RNA one. So when we then kind of go back and what we've learned since then, in just a few short months, we've actually sequenced the DNA, meaning that we've gone through the coronavirus's RNA link by link, think of it as a long chain, and we've identified what each of the nucleic acids are along the way and in what position. We've also learned a lot about the protein coat, the tennis ball, and the little spike proteins. And learning about the spike protein, we've learned about kind of the protein, each of the building blocks of the protein, and the shape of the protein that's on the outside surface of the ball. And we've also learned about, again, just torturing this metaphor as completely as possible, the little fuzzy part of the tennis ball, which are other proteins that are on the surface. And we've learned this in just, a matter of weeks, which was important because that was the way we're able to create sort of a test to test for whether someone had coronavirus or not. and to be able to make sure that we could identify it distinctively from other cousins. Coronavirus, such as like the common cold, which is also a coronavirus that has a lot of little RNA kind of sequences in common with coronavirus, but it's very important to be able to distinguish between the two. So, that was kind of the, extent for like diagnostic tests, but also future tests that we'll talk about in a little while. So the other thing about this, if you think about it, is with the three parts, just the tennis ball, the little flower, and the RNA, viruses really aren't alive in the way that we think of things sort of living. They don't manufacture any energy, they don't create any, they don't respire, they don't actually make anything in particular. They basically are just genetic debris that has a protective shield around it, the tennis ball, that they then use to kind of trick the cells of our bodies into kind of bringing them inside, and it just makes copies of itself. So, quick kind of, run through of just kind of viruses. If possible, try to get a image or diagram of this so that hopefully it makes more sense when we're kind of talking through this. So the second question was, the other day, I smelled smoke from someone walking down the street smoking. I was at least 45 or 30 to 45 feet away in my driveway. Is there any evidence that COVID-19 can travel further in the air if aided by cigarette or vape exhales? And so this is a question that I originally, kind of first time I answered no. And as we do in medicine, I'm going to slightly change my answer this time around and say, like, probably not, but at least not in the kind of what we're thinking about here. So first of all, there's no evidence that cigarettes or vape exhaling will make COVID travel any further than, say, like a cough or sneeze would. But I think the thing that I didn't take into consideration and kind of the first time I answered this question, one is kind of like whether it was wind aided or not. And if the wind was blowing and was blowing the smoke a certain direction in COVID, could it travel further than it would sort of otherwise? And I think it's absolutely yes. First of all, smoking and vaping are also some of the risk factors that increases one's likelihood, if they catch COVID, of having a more serious illness. And I think part of the thinking here is that if you have already partially damaged lungs and you get an infection on top of that, you're apt to may have a more serious kind of clinical course than if you have, pristine, healthy lungs going into that. And I think that's definitely like true. There's also an aspect that we'll talk about later called the ACE2 protein that comes into play here. And we'll kind of revisit this again a little bit later if I don't forget. So, the first part of then starting to unpack this question a little bit is like we talked about earlier, is like in that diagram, the tennis ball, the flower, the DNA, like inside of it, you'll notice there's no visible means of propulsion or movement. So essentially viruses don't move themselves. They're dependent on us to move them throughout the world. And so we either propel them through the air or we carry them. Literally on our bodies, our hands, our fingers, and stuff like that. So the way that we propel them through the air is by either coughing, sneezing, exhaling, or as new research has even shown, even just talking. And so it's all sort of based on this premise of when flying through the air, the virus either flies on a respiratory droplet. Respiratory droplet is just probably a polite way of saying either phlegm, sputum, mucus, spit, or snot. But essentially it's like through one of these mechanisms, we're coughing, we're sneezing, we're that sort of thing, and the COVID virus is actually riding on top of this small little fluid droplet as we force it like through the air. And through some of these mechanisms, we can make the virus or these droplets fly further than others. The other way that they can get into the air is they become sort of separated from the respiratory droplet, and they're just flying on their own, kind of wafting through the air. And so if they waft, they can sometimes stay in the air much, much longer because they're just riding the air currents. And so think of kind of like, hang gliders or paragliders, and they fly around and they hit the updrafts and they can stay like, in the air for, like hours, if not. And so that was one of the really important early kind of considerations. Is this thing attached to these droplets, which actually have weight, and they then fly a certain distance, but because of weight and gravity, they sink and they fall pretty quickly? Or is it through this aerosolized vehicle, which they could, which is how the measles spreads, and would stay airborne much, much longer? So again, this isn't completely settled ground, but the majority of the scientists believe that it's predominantly the respiratory droplet. At least the vast majority of the time. There are ways that you can aerosolize COVID. There's certain medical procedures. There's certain, things like that. So like when patients are getting breathing tubes put in, there's almost certainly like aerosolized COVID in the air, which is why you don't want to be anywhere around that when that's happening. But the good news is that generally it seems that they are sort of attached to these droplets, and that limits like how far they travel. So this is where the 6 foot distancing kind of came from, because it was generally thought that that's about how far that these respiratory droplets, can fly. And so as long as you're kind of outside of that, you should be sort of safe. I think newer evidence suggests that it may be like further than that, but it's still the same general sort of concept, if you will, through that. And I think the important part that this question sort of brought up was the fact that they do travel through the air. They travel different distances. For example, coughing and those type of activities, sneezing and such, probably the droplets travel much further than they would otherwise. Now, for the most part, sneezing hasn't been a symptom associated with COVID. I suppose you could have COVID and allergies, and that way be like sneezing COVID particles or whatever, but that also means you're having a really bad spring as well. So the exhaling sort of tobacco or vaping, think of it as a tweener. It probably travels further than if you're just speaking, but not as far as if you're sneezing, that sort of thing. So a lot of this also then became kind of the basis on what the Center for Disease Control and the World Health Organizations, when they came down and talked about the use of masks. And if you remember, the original recommendation was that we don't need to wear masks unless we're sick. And again, that was kind of the premise that if anybody's sick, we want them to keep the respiratory droplets like literally like, millimeters from their face and not like out in the community, that sort of thing. So we just had to put masks on sick people. Again, the most important thing about masks is you just need to realize is that the masks are to protect, the world from you, not to protect you from the world and that sort of thing. And so basically we put masks on people we thought might be ill, and we should have things sort of covered. One of the odd things that then sort of came along was we started finding all of these patients that were infected with COVID, but they weren't showing any symptoms. So in theory, they were running around the world unknowingly, like infecting others, again, through breathing and, perhaps coughing or whatever, without knowing it. Or we had patients that weren't developing symptoms until really, really late in their clinical course, much longer than common or at least ordinary for these sort of things. And so then we didn't really change our guidance with masks. We just changed the definition of who's sick. And we started to sort of like, let's assume everyone is a asymptomatic spreader of COVID. And so we'll just put a mask on everyone when we go out into the world. And then that way we should be covered. Now, the World Health Organization decided not to recommend everyone wear masks, but they actually didn't disagree with the CDC's premise. They sort of then made their decision based on they didn't feel masks in and of itself was an effective strategy, that you would still need to like socially distance and that sort of stuff as well. So I think this is just an example of two different, clinical bodies looking at the same evidence, coming to the same conclusions, but then making slightly different sort of recommendations, after that. So lastly, if you're wearing a mask, and kind of in this example, for example, let's just say this person who asked the question actually was wearing a mask when they like smelled sort of the smoke. The important thing to realize there is if you can smell either smoke or food cooking, perfume or something while you're wearing a mask, the mask is not protecting you from COVID, right? I mean, all of those little particles that you're breathing in and inhaling are actually, for the most part, much larger than COVID particles and much easier to get around sort of that mask. And so it just kind of hammers home the point that, the typical sort of cloth masks or even like some of the surgical masks or whatever do very little to actually protect you from the outside world. They're more to protect the outside world from you. And I guess just to kind of, when we talk about sort of COVID spreading and this sort of stuff, it's been doing through the mechanisms we just described, it's been doing it effectively well. So on average, each person that has COVID is going on to infect about 5 other people, which is much, much higher than we see with the seasonal flu. It's nowhere near as high as like with measles, but that's why it's been able to sort of spread so fast. So question number 3. If I'm just out exercising in a mostly isolated area, do I need to wear a mask? And again, the answer kind of with this again is probably no, right? Because again, the masks are to protect, the world from you, not that sort of thing as well. So all of the same principles sort of apply. If there's nobody else around, that there's really nobody to protect, from you. Sort of things. Interestingly enough, though, if you are out running if you're running with like somebody else, the studies show you're better off to run sort of side by side, slightly apart, obviously, than for you to be running, say, like, 10, 15 feet or something behind that person, because as you're running, you're actually accelerating into all of the breaths that they exhale. And you're much more likely to be covered with COVID sort of that way by like trailing behind somebody versus if you're at the side and all of your exhaust and coughs and other stuff are just going back behind you where there's not a person. So just go tandem, not sequentially. Okay, question number 4. I had friends and a family who were affected with very similar symptoms as COVID-19 back in December and January. They were not diagnosed at the time. Could this virus have been around much longer than what's being reported in the press? And if so, could we be on the backside of this curve as we know it today? So this is like a great, great question. And, at the time when I first answered this question, I said possibly. And so now I'm going to change my answer, like we do, to, very possibly. And so what's interesting with this question is that, the first case of COVID was kind of identified in China on December 10th. And again, that's like the date that somebody was sick, that we then cultured it, took it back, and, it took about 10 days after kind of they took the initial culture that we identified this as something that was different than coronaviruses that we had seen before. So it's safe to assume that, we didn't catch like the first person who had this, right? This had more than likely been kind of spreading through the community in China for at least a period of time prior to December 10th. And then that was sort of like when we first discovered it. Now, the similar case we discovered in the United States on January 21st. And again, that was in Seattle. And the way that we actually identified it there was we were looking for people that were sick that had recently had family members who had traveled from China. Similarly, it's very likely that that wasn't the only person that either had family members or had actually traveled to China themselves in the United States. And so again, that was just the first case that we happened to find and catch. So again, it's entirely possible that this was in the United States for a period of time before that, and we just happened to discover it then. What's later gone on to kind of support this theory is that we've now found out that we actually have not one strain of COVID, but we actually have 3 strains of COVID. So, which we've named like strain A, strain B, and strain C. Now, strain A is thought to be kind of the original strain of COVID, and this is the one that we think originally originated in the Wuhan area that we first identified back then. So strain B is a daughter of strain A. And there were 2 genetic mutations kind of in the RNA chain that we talked about earlier that changed. And that one is the one, interestingly enough, that was first kind of identified in New York City but was thought to have came there from Europe. Strain A, the original sort of chain, was the strain that predominantly was in South Korea, and then we saw in like the Seattle, Washington sort of area. And then there's a third one that's been in other parts of the world. Again, strain C is a daughter of strain B with like one additional kind of genetic mutation from that. One of the interesting things here, it's just anecdotally looking on the surface, it seems that both the Italy and New York City sort of strain has behaved much differently, at least in terms of the morbidity rates and stuff, than the strain that affected predominantly like South Korea and the Seattle area. But we're really early on in this, and so there'll be more kind of research into that as we go on. So again, great question. Okay, question number 5. Will the virus survive in sub-zero conditions like a freezer? And the answer to this is like probably. In fact, not only will it probably survive, it'll probably thrive. In fact, many labs actually freeze their virus as a way to like store them for like future research and stuff like that. So the other kind of, I guess, where this kind of question opens up, so if they survive cold, what don't they survive? And so when we're talking about this, we're now starting to look, so how do we kill a virus, right? It's kind of an odd question because viruses aren't really alive to start with, but, if we want to inactivate them or disintegrate them or, basically make it so they can't like infect us any further or other people, how do we go sort of about that? So the first kind of way is soap. So if you remember again back to high school biology class, soaps are kind of interesting kind of molecules because they have one end of the soap has an area that is attracted to water, sort of hydrophilic area. The other end of soap molecules have an area that are repelled by water, sort of like hydrophobic sort of areas. And so what are like soaps good for? Soaps are really good at cutting holes, almost like little Ginsu knives, in these type of molecules, which are similar to the tennis ball or the protective membrane, which is kind of a biphospholipid kind of membrane or whatever. So think of these like little knives, like when you're washing your hands, it's like every second you're putting another slice or tearing another hole in the tennis ball. And the reason we wash our hands for 20 to 30 seconds is we think like 20 to, one hole per second, 20 to 30 holes is probably a sufficient number of holes to kind of disrupt this membrane so that then the RNA is then exposed kind of to the environment and it will dry out and disintegrate very rapidly after that. So soap definitely works. So what's another approach to sort of do this? And so now instead of like cutting holes in it, let's try to dissolve it. So what will dissolve the tennis ball? And in this case, it turns out that like alcohol of at least 65% will dissolve the tennis ball. And again, kind of rupture it and inactivate sort of the virus. And so what is alcohol with 65%? So, not by coincidence, most hand sanitizer has at least 65% alcohol in it. And so it's very effective at sort of dissolving or just, that sort of the tennis ball. Other alcohols like Listerine has 65% alcohol, but you still shouldn't drink it. The alcohols that don't have 65% are your typical, vodka, bourbons, those sort of things peak out at about 40 to 50% or whatever. So you're going to have to use something stronger than those in order to kind of dissolve the tennis ball. And then the last way is like, how can we melt the tennis ball? And the tennis ball is very easily melted just with heat. So again, heat in terms of like, cooking your foods, kind of boiling or microwave, that sort of stuff, any potential virus will just be immediately sort of dissolved or melted, if you will. Other ways when you're like washing your clothing, like warm water, hot water, and then dry it on the heat cycle if possible. All of those are very effective ways at kind of like, killing or inactivating sort of this virus. So next question, why is COVID only attacking old people or people who are already sick? So again, this like super great question. Probably one of the most important things just from this question is just because mostly older people are getting sick doesn't mean that younger people are safe. There are a lot of younger people who are also getting sick. So just because the chances are that it's more likely for someone else, if you get it, it's 100% for you. So it doesn't even matter that it was unlikely to occur. But there's kind of a misnomer. I think when we first started talking about this, we put so much emphasis on the at-risk kind of populations that we may have been unintentionally sending the wrong message to the younger people that they were safe from this. And while they're safer, that they're not completely safe. So they just need to act accordingly, that sort of thing. To me, one of the more fascinating things kind of about this has been sort of the role of sort of what's now become as the ACE2 inhibitor. So ACE stands for angiotensin converting enzyme, and the two is, kind of receptor. And these are receptors that are on certain cells of the body. And so what's fascinating about these is they actually play many different roles. But one of the most important roles that they play is they serve as the lock um, on the doorways into the cells of our body. And so one of the things that we've learned is that, we talked about kind of the little flour protein, or the thing that looks like a mung bean. COVID uses that protein as sort of the key, if you will. And that key is very specific for these ACE2 receptors that are on like the human cells of the body. And so when COVID comes in contact with these cells that has an ACE2 receptor, those are the cells that COVID's able to actually get into the cell from. And it's very specific. And so now we have kind of a key, and we have like a doorway, and we have a lock on the door that, the virus is able to sort of open up. So now the really important question is like, first of all, where are these receptors in the body? And, interestingly enough, they happen to be in the area where most COVID symptoms occur. So the majority of ACE2 receptors are in the lungs, and COVID is primarily a respiratory sort of like illness. However, some of us have ACE2 receptors in our gastrointestinal tract. And some people with COVID present, at least early on, or predominantly with more GI symptoms. And others of us have ACE2 receptors kind of in our nose and nasal pharynx and that sort of thing. And some people present with symptoms of either loss of smell or loss of taste and stuff. So as we look at kind of the parts of the body that ACE2 receptors are associated with, it matches up really well with the symptoms that COVID, patients are presenting with. So perhaps even more interesting is who has kind of ACE2 receptors. And, ACE2 receptors are primarily found in the elderly, and they're much more common in patients that have other chronic illnesses like chronic heart disease, chronic liver disease, chronic kidney disease, diabetes, high blood pressure, and people who are overweight. Have a lot of ACE2 receptors. The reason they have a lot of ACE2 receptors is that ACE2 actually plays a protective role in the body. They help reduce inflammation, and all of these chronic conditions are associated with increased levels of inflammation in the body. And because of that increased inflammation, the body actually manufactures more of these as kind of a protective mechanism to help sort of mitigate or manage the inflammation levels sort of like in the body. So of course, these are going to be the populations where the body has upregulated, it's called, these particular receptors. It's just really unfortunate that this is the key, the lock, if you will, on the door that the COVID virus is being able to get into. So the hypothesis here is that by having more of these receptors, those individuals are more susceptible to these, meaning that this is the only way it can get into the body, and you have lots of entry sites or portals able to get into the body. And if they're able to get in more effectively, there's maybe a dose dependent phenomena where more of these COVIDs will get in. It will create a much serious clinical illness than if you only have a few of these receptors and you're not able to get into the body as well. So again, this isn't, this is just a hypothesis. This hasn't been proven. It hasn't been settled science, but it just, the correlation or the association, if you will, between sort of, who COVID is picking on and the people that have more of these. And we do know for a fact that this is actually how COVID gains access. And we know that young children often don't have very many of these. Younger people have fewer of these and that sort of thing. So it seems like, if not, it's a helpful way to think about it, and it helps us kind of anticipate who might be getting affected next and how they might being affected. So it's a useful kind of mental model, or just helpful way, I think, to kind of think about it. So, and we'll talk a little bit more, I think, a little bit later about kind of the implications around the ACE2, and if you inactivate its anti-inflammatory response, that also may help explain some of the other things we're seeing with COVID. So, question 7: has there been a fast-tracked vaccine process that could make one available before next year? And this is probably no. Because I think, in terms of like testing a vaccine or creating a vaccine, the good news is the initial sequencing of COVID happened in like record time. I mean, super, super fast, just like a number of weeks. I think University of Texas, Austin was one of the first to sort of sequence this, and they did it really fast. And so that gave us a really good head start as to creating a vaccine because we knew the genetic fingerprint. Even once you create a vaccine, you have to go through a series of kind of steps, and each of these steps takes time. And the first step is just proving that the virus or the vaccine is safe, right? So this means you have to give the vaccine to humans, and you have to wait and make sure that it doesn't make them sick or cause any other serious kind of consequences, right? Because it needs to be something that you can administer, in large volumes to the population, that sort of thing. So the first part is just a safety. You don't even have to prove that it works. You just have to prove that it doesn't hurt people, or hopefully not kill people. And that you just have to wait a while after you give it to them to make sure nobody's getting sick. So phase 2 is effectiveness. Not only do you have to not injure people, you have to actually prove that you're sort of good at doing whatever it is that you're trying to do. And in this case, prevent people from catching COVID. And that takes even a little bit longer, right? Because you have to give it to enough people, and then you have to wait and give it enough time to go by to make sure that, statistically significant more of the people that you gave it to didn't get sick than people who went on and got sick on their own was sort of out of it. And so those are things that it's hard to just kind of rush kind of that time frame as well. Generally, there's a third phase where you actually have to prove that you're better than something that we already have. In this case, we don't have anything, so we can kind of skip that step. But now we just have to manufacture it in large enough quantities that we can sort of get it out. And so I think the earliest is we're looking at is going to be, maybe, spring, early summer next year. And that's if everything goes perfect and we get through these steps as fast as we can. So vaccine's going to be a little ways off. So 8, is hydroxychloroquine safe to take to lessen my risk of contracting COVID-19? So hydroxychloroquine is a really interesting drug. It's a drug that's been around for a long time. It's used for very specific things. It's not used as common as it used to be because there's newer, safer drugs, sort of available. But this is kind of another one of those categories where there's a lot of I guess sort of pharmacologic and biochemical reasons why hydroxychloroquine could be effective at sort of both, decreasing COVID's ability to enter cells. So it kind of interacts with kind of that flower and the ACE2 receptor sort of thing there. And also its ability to sort of replicate and stuff. So, it's a medically good idea that's interesting. However, there's a lot of medically good ideas that work in the petri dish. They may work in mice, but they don't then work in humans and stuff. So that's one of the things we need to kind of test here. The other thing with hydroxychloroquine is it has a very high side effect profile. It can cause some really serious side effects in people who take it. And so we're now in kind of one of these where we're trying to weigh the benefits of the risks. And we know that there's a lot of known risks, and we haven't proven that there's any benefit yet. So it's kind of a no-win situation, at least as for the time being. Now, it means we should study it, but it doesn't mean that we should just go out and just start taking this, just because of the unknown. And this is a case where doctors who are administering hydroxychloroquine may be overestimating sort of its benefit. When we look at like, between 3 and 5% of COVID patients will require sort of ICU and ventilation, which means that, anywhere between 93 and 95% of COVID patients will eventually get better on their own. They may have a really difficult course, but they will get better on their own. So when we give hydroxychloroquine and then the patient gets better, it's hard for us to really tell whether the patient, would have got better anyway. As doctors like to think that it's something we did that helped the patient get better and stuff. So we got to eliminate that type of bias and do this thing in a real rigorous scientific way or whatever, or we'll just be talking ourselves into some benefit that may not actually be there. So question 9, do antivirals help at all? So I think antivirals are very promising. Right now we have no proven, similar to the hydroxychloroquine, evidence that any of the antivirals help. Again, that there's medically good ideas here and reasons why they might work. I think the antivirals that may have the best chance will be some of maybe not like the Tamiflu, the thing that works for the seasonal flu, that kind of decreases the symptoms and shortens the course slightly type things, but we may be looking at some of the ones that are used kind of more for like HIV or even some of the Ebola and those type of things as well. So I think they all kind of interfere with viruses in slightly different ways. They either mess with their protein code or their surface proteins or the RNA replication, those sort of things. But again, we just don't have any evidence right now that any of them are, clearly effective. So number 10, how much would you emphasize the importance of immune boosting support like vitamin C or zinc as an effective preventive measure? And so this is a great question. And again, I think it's a great question because, well, first of all, these are kind of different therapies that are being administered in, large hospital trials. Today, I know that Northwell and other areas are using large doses of intravenous vitamin C and other places are using zinc or whatever. But I think one of the things here is that they're not being used so much to boost the immune system. One of the things that we've discovered in kind of COVID is it's generally or oftentimes not that we have an inadequate immune response. It's that we have a bit of kind of a runaway immune response that either is, much greater than is actually necessary or doesn't stop when it should. And so it continues to go on. So at some point, the actual biggest issue is no longer the COVID, it's the body's reaction to the COVID, and a thing which is called kind of like cytokine storm, or you may hear a little bit about this or whatever. So there's actually very specific treatment protocols now, how to get the immune system to calm back down again when enough is enough sort of thing. And again, this is another area where this ACE2 receptor that we talked about comes into play. One of the other, we talked about ACE2 as kind of having this anti-inflammatory role. Originally, it then is one of the things that signals to the immune system and, to keep it sort of in check. And that's actually the beneficial role that it's playing in the bodies of the people with these chronic illnesses and stuff. It's thought perhaps that when the COVID, virus, via its, flower protein sort of attaches to and interacts with the ACE2 receptor, that it damages its immune modulation function, which at that point, essentially, it sort of like inactivates the brakes, if you will, on the immune system. And that may be part of why we're seeing these kind of runaway responses, because we've lost our controlling mechanism, for the virus and stuff. So again, more of this is just, speculation, but it seems like a lot is sort of lining up kind of around this ACE2, receptor. So lastly, the thing I just would like to kind of take a little bit of time with is discussing sort of immunity and immune sort of tests, if you will. And again, I think, the main question, or one of the questions that comes out of this that everybody wants to know, is if I have COVID, can I catch it again? or am I going to be protected for either a while or lifetime or that like sort of things. Um, and also, if that's true, how do we test for that? And so there's a number of kind of blood tests that have kind of been in the work, and they all sort of focus on sort of antibodies. Um, the one thing with this is like all immune tests are antibodies test, but not all antibodies test are immune test, meaning that, many of the tests that just tell us that the body's been sort of exposed to the virus before. So if we kind of walk through this step by step, our body forms, three different types of antibodies. Um, Antibodies IgM, IgG, and IgA. So I remember this by the British sports car, the MGA, which came before the MGB. But the first on the scene are these like IgM sort of molecules, and that's kind of our body's initial response whenever something's going wrong as IgM antibodies go. And they're essentially signaling cells. So they signal, they say, yeah, I found something, and they call like the rest of the sort of immune system. And so that's sort of their role. So when you find IgM antibodies specific for COVID, first of all, is your body has seen COVID. Doesn't really tell you the rest of the story or what sort of happened after that, other than, it's identified that. So the next sort of thing that happens, and this is the really cool part, is like these IgM antibodies kind of do a transformer sort of thing, and they shapeshift. And what they do is the IgM unfolds these things that looks kind of like the solar panels on the space station. And essentially what they do is they cover up the flower proteins, and they kind of block other things from attaching to sort of those. So they're kind of temporarily holding the fort. They're not really doing much other than just getting in the way. But that's kind of a later stage, and that kind of is your kind of clue that, that a little bit more has gone on rather than just kind of a casual, kind of encounter with a COVID sort of molecule. Um, Lastly, IgA is kind of the solution that we're looking for. So IgA antibodies, it's almost like they take a, picture, of the flower protein, the COVID, virus, that sort of things. And they send it to the immune system. So it's almost, think of it as kind of like the FBI's like most wanted list. And so they actually get this kind of, actually very detailed, set of specifications of both molecules and molecular structure and stuff, and distribute it. So and each of the body then kind of keeps these on hand. So if it ever sees this again, it identifies it and attacks immediately. And that's when you have immunity. So when you have evidence of both of IgA antibodies kind of to COVID in sufficient quantities, right? Because we measure these things kind of by, we call them titers, but it's basically like counts. It's like how big is your army sort of thing. And so when you have a lot of these sort of in your system kind of going around, then you're going to be protected from catching this again. I mention this is because the immunity test is being sort of talked about as one of these critical things that we need to have before we open up the country again and kind of, things go back to sort of normal. We need to be able to diagnose, but we need to be able to like test people and figure out that they're like immune to it. They're talking about sort of passports and things that shows that you're safe to let into the country because you're not going to get this, and you're not going to spread it. They're talking about healthcare workers on the front lines. If we could find out who's immune, they could work with like very little or no kind of protective equipment safely. So there's a lot of implications to this, and a lot of just being able to kind of measure, when do we actually have what we call herd immunity in the population that makes it very difficult for a virus to transmit because most of the people are resistant to it. That sort of thing. So like each person, instead of infecting 5 people, each person is now infecting less than 1 person, which means that this will essentially burn out at its own time sort of thing. There's been over 70 of these immune tests either released onto the market or are coming onto the market literally in like this week or next or that sort of thing. One of them has been approved by the FDA. The other 69 have been approved based on kind of what these, EUA or emergency use, approvals, which means that we don't have time to test all these things. So we're going to let these kind of onto the market. The important thing here is just to be careful with how we sort of interpret these. Like today, every test um, is just the IgM or the IgG, right? So it's kind of either the initial one or the one that does the transformer thing. We don't really have any of the tests that sort of distinguish between we've now actually have a stored, immune, response, at the ready sort of thing. So, really they tell us nothing more other than you've been exposed, but there's no guarantees that things are going to be different sort of like in the future. So we just need to interpret those. Also, many of these tests, if they are just some of the IgM, IgG, they may not be able to really distinguish between COVID and the common cold. Again, they're cousins. They're like very closely related. They have similar, kind of the flower, the S protein sort of configurations and things like that. And similarly, they may not be able to distinguish between other like coronaviruses like SARS and MERS and those sort of SARS and MERS, those sort of things from the past and stuff. So again, we just need to kind of proceed carefully and just realize that at least for the time being, these may have significant limitations and stuff. So we just need to be careful, especially as we're trying to like safely, reopen the country type thing. So again, that's all of the questions I have for now. I want to thank you, for joining us today. For more information about you can text Corona to 83973, and please be safe out there. Thanks.